Hello friends, today we are going to discuss about important molecular tools used in genetic engineering. Enzymes and vectors used in genetic engineering. Enzymes and vectors used in genetic engineering. Before we go into the exact topic, we have to learn about the basic protocol of genetic engineering. How and what is the crux point for the successful revolutionization of genetic engineering? It's about you have to generate a particular DNA and identify our desired DNA fragment and cut out the same thereby restriction endonucleases and identify the vector, say plasmid, then cut down at a particular location, then incorporate the desired foreign fragment into the plasmid. Then we create a recombinant plasmid, that means recombinant vector. Then you have to include the same into a host cell, then allow it for multiplication, that means duplication, and identify the specific clone by means of appropriate marker and the end product. It's about the basic protocol. Okay, for example, this is a foreign DNA, then this is our desired fragment, desired DNA fragment. Says it is nothing but gene. After that, restriction endonucleases. Restriction, subject the same to restriction endonucleases. Then you will get the particular small fragment. And the other way, this is a plasmid. Subject the same to restriction endonucleases. Restriction endonucleases. Then we'll get open cut, that means it's a plasmid. We'll get open cut plasmid, open cut plasmid. Then through ligase join the two. Say for instance that creates a recombinant plasmid, then include in the appropriate host cell for duplication. That means you have to allow it for multiplication and specific identification of the clone, identification. Then finally you will get our desired end products. This is the plasmid and this is the end product. It's about the basic protocol in genetic engineering. Just I generate a DNA, a particular DNA from a source, then we would like to identify our desired DNA fragment, that means which is nothing but a gene of interest, and cut down the same to restriction endonucleases. Then identify the clone, that means is a plasmid, which is used as a vector. Then cut down the same at specific sequence so that the desired DNA fragment and the plasmid may give rise to a recombinant plasmid. Then identify the host to include, that means introduce the recombinant plasmid to the host, allow it for multiplication, and identify the specific clone carrying our desired DNA fragment and the end product, that means the protein encoding by the concerned gene. It's about the basic protocol of genetic engineering. Here, which are the most important steps or which are important molecular tools without which we cannot expect the successful revolutionization of genetic engineering? Number one, the restriction endonucleases. Number two, the vectors. Last one is here vectors. Because of these two tools, molecular tools, the entire regime of recombinant DNA technology, what we call genetic engineering, relies upon. So today we are going to discuss about that. Okay, so that's how, that means the protocol, for a protocol of genetic engineering, the basic is these two, enzymes and vectors. Okay, the most important enzymes we are going to discuss used in recombinant DNA technology is restriction endonucleases. Restriction endonucleases. The word it implied, restriction. Before, first we will discuss, then we can have a schematic representation. How and from where the word derived. Restriction means for the first time these are the enzymes which are otherwise called DNA cutting enzymes identified originally in Escherichia coli, and these enzymes happen to restrict the replication of a virus, say bacteriophage. That means they restrict the viral replication. Restriction of viral replication. How? Simply by cutting down the phase DNA. That is how the word derived. Restriction, restricting the viral replication endonucleases by cutting down the phase DNA. Okay. And most important point is they specifically identify a particular sequence in DNA so that they can cut. There are two kinds of cut. One staggered cut and two straight cuts. Staggered cut leads to sticky ends or cohesive ends, whereas straight cut leads to blunt ends. And these staggered cuts that leads to sticky ends or cohesive ends because of this specific recognition of restricted endonucleases leads to successful human genomic or genetic manipulation experiments. Say for example, we are conducting a gene manipulation experiment. We would like to isolate a gene of interest, and incorporate the same into plasmid, and we want to get multiple copies. There is a specific protocol to cut down the sequence. Say for example, with the help of restriction endonucleases, which cut along that means staggered cut that leads to creation of sticky ends, so that the appropriate, say for example, we have isolated same, and the complementarity through appropriate another sticky end will be introduced, ultimately creating a recombination, a recombined product. That means recombination is the basis for RNA technology, and this recombination is possible because of restriction endonucleases, because it specifically cuts the DNA at specific sequences. Okay, this is about the basis. How? I will tell you. For example, they take one important example. Restriction endonuclease, what we call EcoR1. Here, before we go into the mechanism how it works, we have to learn about certain basics. E stands for the genera Escherichia coli, Escherichia. Co stands for coli. R stands for RY13. That means the strain. One stand for order of discovery. Order of discovery. That means Escherichia coli restriction endonuclease 1, it is the first ever restriction endonuclease reported. That's the meaning. Okay. And how it works. For example, I told you already, they specifically identify the DNA, what we call recognition sequence. Specific recognition sequence. Each and every restriction endonuclease, that means there are different restriction endonucleases, and each and every type had its own recognition sequence. And the common point is mostly the palindrome sequence, what we call palindrome sequence. Cutting across the strands is most important here. Cutting across the strand leads to generation of sticky, and I told you that across is most important. Okay. Palindrome sequence. Let us see how it works. For example, there is one enzyme, one restriction enzyme called EcoR1. Okay. This is one of the, it is a restriction, that means recognition sequences G, A, T, T, C. This is the recognition sequence of E. coli. I don't know what is palindrome sequence. Palindrome sequence are the sequences that read same in one particular direction in both strands. For example, G, A, T, T, C. That means here it both reads same 5' to 3' and same in 5' to 3'. They are called palindrome sequences. And I do know the restriction sequence size is generally around 4 to 8. Okay, and this is a restriction sequence of E. coli. Specifically, it will cut down here and here between A and G and A and G. That I told already. It formed two cuts. What were across the cuts? Here the kind of cut is staggered cut. Staggered cut. What are staggered cut? That leads to sticky ends, otherwise called cohesive ends. You know how it works? Say the next step after we get the end products, say AA, T, T, C, then G, and G, C, T, T, A, A. These are what we call sticky ends. What are sticky ends? They contain an unpaired overhangs both at 5 prime, 3 prime or 3 prime to 5 prime. These are called unpaired sequences. You know, unpaired overhangs, where there is an appropriate complementary sticky ends will come and bind. That's very important. I told you already. This staggered cuts directional leads to sticky ends or cohesive ends is a basis for recombinant DNA technology, because it's specifically added with the recognition sequence, which is of 4 to 8, and this is the recognition sequence of echoing. Then appropriately they form complementary base pairing later onwards. So this is how we can establish a recombinant product. Just imagine there is no possibility of a staggered and sticky ends by restriction, and generally most of the restriction nucleases they produce sticky ends by staggered cuts. Another one is called what blunt ends, what we call, for example, you know, so I do straight cuts. Here staggered means across, you know, there is a particular direction, but a straight cut is different. That leads to blunt ends. Blunt ends means there are no unpaired or overhangs, that means unpaired bases. Say this is T, T, C, G, G, and this is same A, G, C, C. Okay. The straight cuts are restriction only cut down here, and that leads to simply T, T, C, A, G plus G, G, C, C. That's it. There are no, that means straight, the word it implies, straight will cut, it's a blunt, there is a dead end. There are no unpaired bases which allows the complementary stick ends. But the thing is, most of the restriction endonucleases cuts, the direction of the cut, what we call pattern or the mechanism of cleavage is staggered. This is very important, okay? And for ecolysis, we will take another example. You know, I told you already, the naming is also very important. Say hen 3. Hen, H means hemophilus. Hemophilus. In means influenza. Influenza. D means strain. RD. This strain, name of the strain, and discovery of restriction endonucleases. That's the word. That means this has been the name it implied. It has been isolated from a Haemophilus influenza of strain D. That means RD, and is third in the order of discovery. That's it. And how it works, you know. This is the recognition sequence of, you know, hen 3. The same, just like E. coli, one will form here and another will form here. Then that leads to two kinds of sticky ends. Then another one. So these are the sticky ends. Sticky or cohesive ends. This is very important. So this is the reading sequence. This is what we call recognition sequence. recognition sequence of hint 3. So this is how the issue ends. That means the point of action is very specific. They have to read, that means they have to recognize a particular sequence, and at a particular defined site they will cut on it. They will make a cut, one here and one here, a full palindromic sequence. This is how the issue ends, and this is about restriction endonuclease activity. Then one more important enzyme connected with the molecular tools of restriction endonucleases are ligases. In previous classes we have discussed at length about ligases, no? DNA ligases. We knew that DNA has been stabilized by hydrogen bonds, say ATCG, 2 hydrogen bonds between adenine and thymine and 3 between cytosine and guanine, but they are not strong enough, which makes DNA a stronger element. Here phosphodiester, what we call phosphate backbone. This phosphate backbone means the phosphate group of 5th prime carbon group of 1 deoxyribose with that of the hydroxyl group of the 3 prime carbon of the next deoxyribose and another deoxyribose are the elements, participating elements in the phosphodiester bond is a covalent bond. Because of this phosphate backbone only the DNA able to establish its standard structure. So, and who established and who participates this phosphodiester bond? This DNA ligases. The point is clear. They play an important role in DNA repair mechanisms. Okay, how, it's a simple phenomenon. For example, this is a 5th prime and phosphate group. It's establishing phosphodiester backbone for stability, and this is 3 prime hydroxyl group. Okay, then you DNA ligase the hydroxyl, then we can expect the phosphodiester bond between this. Must be got simply for representation I am giving phosphodiester bonds. Phosphodiester bonds. This is how the issue ends. I told you already in the beginning and regarding the basic protocol. After the restriction endonuclease cut down, we should combine the different DNA fragments, one the desired DNA fragment and one the plasmid. And who will combine this? That's the DNA ligase. How? Simply they form phosphodiester bonds where simply the 5th phosphate group of 5th carbon of 1 deoxyribose will have the hydroxyl group of 3rd carbon of another deoxyribose. That means it is clear that one is specific destruction and one way specific construction. That means the entire dependence of recombinant DNA technology is completely on these 2 important enzymes. Of course, I'm not saying that there are not other important enzymes, but these are very important. Restriction endonucleases and the DNA ligases. Apart from these, we have other enzymes like alkaline phosphatase. They remove phosphate group from 5 prime ends in both DNA and RNA, and we have DNA polymerase 1 synthesizes DNA, based on complementarity. Then we have exonucleases 3 removes nucleotides at 3 prime end. We have lambda exonucleases which remove nucleotides at 5 prime end. We have reverse transcriptase that synthesize DNA from RNA. These are different enzymes. Apart from these two enzymes, there are different. Apart from these, there are still more enzymes like RNAs, DNAs. There are specific enzymes which have specific role, and these are some of the important enzymes. Okay, so restriction endonucleases and ligases, DNA ligases are very important enzymes, and other than these are the important enzymes. Then the next important molecular tool are vectors. Vectors used in recombinant DNA technology. Before that, I told already, we require host, what we call cloning factories. Host. Simply cloning factories. Say, in case of prokaryotes, the most commonly used are bacterial groups, bacterial hosts like E. coli, Bacillus subtilis, Streptomyces species. Whereas in case of eukaryotes, we have different things like fungi, Saccharomyces cerevisiae, and animal cells, insect cells, oocytes, mammalian cells, and even enter whole organism and plant cells, protoplast, the intact cells, even whole organism. Generally, microorganisms are considered as a preferable host. Why? The simple answer is because of rapid multiplication. But there is a point for concern. There is a drawback when we use prokaryotes. Say, generally E. coli is the most favorite host for most of the genetic engineers because it's a non-pathogen and most rapidly. But there is a drawback. Is it doesn't have machinery, cellular machinery that promotes, that leads to post-translational modifications. No post-translational modification. This is a very important point. Post-translational modifications. Because of post-translational modification, it converts a protein into its active form. Why we require post-translational modification? It's like we have discussed at length. When we discuss about gene expression, translation. There, the post-translation modification, some of the amino acids get trimmed out to get activated form of the protein. And these prokaryote, then prokaryote host doesn't have this machinery. It's a very, very important setback. But mammalian cells, for example, most common used because they have a machinery for post-translational modification. This is very, very important point. This is very important. Since they become important favorites. But still, despite this drawback, despite this setback, still the scientists consider the E. coli is the most favorite agent. It's about host cells, Then the most important, the vectors. So then I told already, host nothing but cloning factories. We want some institution-like thing, a living system or a cell. That's it, where we can include the recombined or chimeric product for multiplication and specific identification of the clone carrying the desired product that leads to our desired product, let's say protein encoding. That's the next step. That means vectors, what we call cloning vehicles. It's about vectors, cloning vehicles. What makes a vector used in recombinant DNA technology? Cloning vehicle. The vector should be a DNA. It should carry a foreign DNA fragment. And self-replication. These are the most, that means 3 important characters for being a vector. It should be DNA and it should carry a foreign element and foreign DNA fragment and self-replication. Because we must have a number of copies of our desired product. So for that, these are the three important vectors. Then what are the most important vectors? Say you saw about basics, plasmids, phases, cosmids. These are called natural vectors. Apart from these, there are human artificial chromosome. These are called artificial vectors. Say human artificial chromosome, yeast artificial chromosome, bacterial artificial chromosome, bacterial artificial chromosomes. These are called artificial or synthetic DNA fragments. The chromosomes can directly say, because all the chromosomes are. First we will discuss point by point. Plasmids, say PBR 322 or PUC 101. Phases, say lambda phase or M13 phase. Some of the examples I have given here. Say PBR 300 is the most commonly used plasmid, generally used. PBR, this is the most commonly used plasmid. But some people consider it as parent or grandparent of most of the vector, just like forefather. This is very important. There are certain markers associated with what we call tetracycline resistance genes. Tetracycline resistance genes. Ampicillin resistant gene. Ampicillin resistance gene. This is ORI in last ordinary application. Then it is recognition sequence. I told you equal 1 or H in the 3 other regions are also there. This is PBR 322. P stands for plasmid. B stands for Bolivar. R stands for Rodriguez. T stands for the number given by the authors. U stands for here University. C stands for California. Why I have given this? There is a specific nomenclature pattern. The P stands for in smaller cases. B are the authors. That means the workers who have identified or worked out on this plasmid. Say Bolivar and Rodriguez are the who discovered this PBR 322. And 322 they have given the number 322. But as PUC, sometimes we can name after the institutional name. Say plasmid of University of California and the number accordingly. Okay. The most important plasmid is PBR 322. It contains two different genes, ampicillin resistant genes, tetracycline resistant genes. I told you already. There is specific sequence, what we call recognition sequence for restriction endonucleases. If we are using equal 1, it will cut down here, so that the foreign DNA fragment will attach here by through DNA ligase, forming a recombinant plasmid, then including the host cell. That's the thing. If we are using Hen3, it will cut down the specific length. Say, for example, AAG CTTT. AAG CTT is the recognition sequence for Hen3, and GAATTC is the recognition sequence for E. coli. AAG CTT for Hen3. So accordingly, the cuts will be, that means staggered cuts will stick in. That's it. Okay. And this is, I don't know, the most important point is this is the most common used plasmid. Whereas apart from these, we also have phases. Phases, what is the distinction? That means, what is the important feature of phases? Here, the most common is the lambda phase and M13 phase. They can carry larger DNA fragment when compared to phases. Larger DNA fragment when compared to phases. This is very important point. Whereas cosmids, they are combined product of plasmids and lambda phase. They also can carry larger DNA fragments. So since phases carry larger DNA fragment, they are most commonly used in human genetic related experiments. Whereas with respect to cosmids, they carry larger fragments. Larger fragments compared to plasmids. Very important. And they have a combined features of plasmid phases. For example, we have constructed a cosmid, say, from lambda phase and a plasmid. Lambda, just like what is a phase character? It can pack like a phase and inject into the host cell by phase, whereas replication by means of plasmid. That's it, a combined product of the two. Whereas, why we want artificial chromosomes? Nowadays, we are also using artificial chromosomes as vectors because of larger DNA size. Say the foreign DNA fragment is say somewhere around 300 kb, sometimes even 2000 kb. I told you already, 1000 base space equal to 1 kilo base pair, 1000 kilo base space equal to 1 mega base pair, 1000 mega base space equal to 1 giga base pair. So carrying larger DNA fragments of huge size required specially prepared vectors. Then the came into case human artificial chromosome, what we call microchromosome. A microchromosome which can be generally used in gene therapies. This is very important. Human artificial chromosomes used in gene therapies. It can carry a larger DNA fragment, but yeast artificial chromosome, so far the best one, which can carry a DNA fragment, foreign DNA fragment of size 200 to 2000 kilo base pairs. This is a very important point. Yeast artificial chromosome can carry a DNA fragment. That means depending on, we can say depending on the size of the foreign DNA or the desired DNA fragment, we can pick up the vector appropriately. That's it. If we are dealing with larger DNA fragment, deal with ACC, yeast artificial chromosome, simply called yeast artificial chromosome. It is called human artificial chromosome. It is called bacterial artificial chromosome made of F plasmid. That means I can say simply just like back means what is the distinction between the bacterial artificial chromosome with the normal plasmids? They are made up from F plasmids, and their size is relatively large. That means they can carry larger DNA fragments. That means human artificial chromosome, yeast artificial chromosomes, and back relatively can carry larger DNA fragments. This is very important. That is why we are depending on this. Just like in mammalian cells, we require there is a machinery for post-translational modification. That's why we use mammalian cells, like insect cells, the oocytes, and even entire whole organism. That means animal cells, particularly the mammalian cells. These mammalian cells have a machinery for post-translational modification. That means in the animal cell categories. That's why we are using this for to convert the protein into active form. This is very important. But in case of prokaryotic hosts like E. coli, they doesn't have post-translational modification. That is why they distinguish. So the most important point is, I'm not going to deep into the basic we have learned, but the distinguishing features we have to understand at high advanced levels, because in examination point of view they will ask on this line, what is the significance of yeast artificial chromosome YAC? Because it can carry a larger DNA fragment of 200 to 2000 kb. So our focus is specific identification of the key markers. And why PBR is common? Because and how it can identify. There are DNA markers like tetracycline and ampicillin. They are called DNA markers. With the help of the same, we can identify in the clone. So specific identification of the topic and accordingly what to learn. Okay. And it's about tomorrow today's class. Tomorrow we will discuss about some other topic. Okay.